right, welcome to STEMpaths. I am Dr. Thornton Larson, and here we have students unpacking papers, study paths, and the people behind them. Today we have our guest hosts, Alfie and Emily, and we have with us our guest, Dr. Patrick Mulcrone, who's got to say a quick disclosure because he now works for the company Lilly, which is not necessarily associated with this paper. Go ahead, Patrick, really quick.
Thank you, Thornton. So the content and views expressed are my own and do not represent the views of my employer, Eli Lilly and Company.
All right. With that, Alfie, Emily, would you want to take it away, reintroduce yourselves, and then get into a quick conversation?
Yes, sir. So, hello, my name is Alpheaus Cann, and I'm a biology major. And a quick little summary about your paper was you saw that researchers tested 5 different AAV8 vector designs in healthy and hemophilia B mice to evaluate long-term safety, and no liver tumors were detected in any group, supporting that AAV is not inherently tumorigenic. However, vectors were incomplete genomes where strong CB promoters caused noticeable liver and spleen abnormalities, including increased collagen deposition and immune activation. In contrast, the TTR-HFix vector produced minimal pathology and showed the best safety profile. Overall, AAV safety varies significantly with vector design, not with the AAV platform itself. And then if Emily wanted to add anything.
Yeah. Hi, my name is Emily Martinez. My major is biology. I give a quick summary of what I commentation in the article, and which is the study shows that high dose rAAV vectors do not cause liver cancer in the mouse, but do produce liver and spleen. And those effects depend strongly on the vector's genomic design. Vectors with incomplete genome or strong unique promoters cause the most pathology, but the therapeutic design produce minimal advanced effects. Did we miss anything in our summary?
No, I think that's a pretty comprehensive and good overview of the study. Full disclosure, I did have to reread it this week. It's been a minute since I've looked at this paper. But yeah, so the story behind this manuscript. So I had finished so I finished my PhD in cancer biology in 2017. And so typically if you're someone who gets that type of a degree and you want to stay in academic research, so you want to run a lab and you want to teach students at a college, you typically go and do more research after you get your degree. So they're called postdoctoral research internships, fellows, experiences. And so I had completed one postdoc research experience in a completely separate field from 2017 to 2020. And so when I joined this lab, which is the gene therapy lab, this was the first manuscript that I had published with this group with my mentor, Weidong Xiao, who is the person you initially emailed and he connected us to have this great conversation. So this paper, in some respects, I think is a really good one for a discussion like this with students. Biology classes have changed, I'm assuming, dramatically since I was an undergrad way back in 2004 to 2008. But what is nice about this paper is that I think it gives a very high level, overarching kind of assessment of what you can see in your models and kind of big picture what you can observe versus getting into molecular pathways or specific proteins or specific cells. This kind of shows, kind of relatively speaking, large changes or large differences that can be seen based on, like you all are saying, the design of these recombinant adeno-associated viral vectors. And there's definitely, rereading it, there's definitely some things I wish I would have done differently and redesigned, and there's definitely some limitations. But I think overall, it's a kind of a very good, just almost an educational paper versus, something that is technically, very groundbreaking or, changes the field a little bit.
Sir. And I had to read it a couple times. It was like a very interesting paper, but a lot of points were kind of confusing to me. So like for listeners who may not be like familiar with gene therapy, like how would you explain AAV vectors and why they're so widely used?
Yeah, that's a great question. So, some of you probably read this paper, I was like that when I first joined this lab back in 2020. I knew gene therapy was, that's where everything's going. Just kind of taking a step back at the moment now. I truly felt that being in this lab, truly felt it after I finished this lab, and I still believe it. Gene therapy is the way of the future of targeting, rare diseases, cancer, diabetes. That's what I believe. I see it I saw it in the literature back then, and I still kind of see it now. So with adeno-associated viruses specifically, so these are non-pathogenic viruses that are being used for, the main reasons are they are not as immunogenic. So what that means is that if you give someone, let's say someone gets a treatment and it's an antibody as their target, and you get some sort of reaction, so you have an allergic reaction, or your immune system gets activated because of that type of medicine. What these AAVs have shown compared to some other types of viruses like lentiviruses or adenoviruses is that they cause a less severe immune response. So that's one thing that's really great about these. And the second point, one of the other points about why these are great is that they have a long-term effect. So you think about someone that has to take a pill every day to get the effect for their medicine to keep themselves healthy. I have a digestive disorder. I take 4 pills a day. It's working. It's keeping me in check. But what these AAVs might do for patients on the road is that they get maybe 2 injections a year, 2 treatments a year, and they are completely either, stable disease or in remission. So that's kind of why these are so they have the potential to be so powerful. And you see that there are several of these types of AAVs that are already approved for various diseases. Hemophilia A is one of them. I have my little list, have my little cheat sheet here because I had to kind of go back and relook. But there's, you have retinitis pigmentosa, you have spinal muscular atrophy, you have all these varying diseases where AAV technology is already approved and having therapeutic benefit for these people with these diseases. So that's kind of the why they're a very, it's a very interesting field to be part of, and it has so much potential. And kind of jumping off of this, we'll get back to David, but from this manuscript, I really wanted to investigate whether these AAVs could be used to target cancers or different cancers that maybe were more difficult to target with standard medicines like small molecule inhibitors or antibodies or even immunotherapy, which is another arm of cancer treatment that's really revolutionized the field. And so that was this was that first jumping off paper where Dr. Shao said, get your feet wet, understand the biology, understand the science, do these experiments, lean into your experience. I had a lot of in vivo animal experience in my training. So he said, lean into that, do a manuscript like that, and then we can kind of start to design some studies where cancer is more of a focus.
And then a quick little follow-up question. What inspired you to compare these specific vector designs?
Yeah, that's another really good question. The way that we describe it in the paper is you have this idea of the word strong and strong promoter. Strong is kind of a synonym in this case for not necessarily non-specific. So if you have, the CB stands for chicken beta actin. So it's, actin is a very, it's a common protein. It's used as loading controls for Western blots and things like that. So it's a very, it's a very, not superfluous, but, common protein. So that promoter is just what's called a strong promoter, where it actives, it transcribes genes of interest in these recombinant AAV vectors, versus if you have something which is the TTR, which is the transthyretin promoter, which is a protein that helps that AAV vector target specific cells. So the TTR, it's not necessarily a strong promoter, it's more of a specific promoter because you want to have an effect that is very targeted or very localized or very precise. And so, because you run the risk of, if you use something like a strong promoter, let's say you inject animals in this case with a strong promoter, that doesn't help with guiding where that virus goes, what tissue or what cells it goes to. You can get an effect in an organ that you don't want to target. It's a similar type of thing. Think of like chemotherapy, right? Chemotherapy you give to the whole body. So that's why when people, the tumor maybe gets affected, but people lose their hair, people have digestive issues because it targets rapidly dividing cells. It's not specifically targeting a cancer. It's targeting all rapidly dividing cells. It's kind of the same thing with this. So a CB promoter, chicken beta actin promoter, would maybe give you a robust response, but it's not going to give you necessarily the most specific targeted response that you might want with your vector. So hence, that's why we compared the two. And with the TTR HFIX, which is that was a design that is effective for targeting hemophilia. So hemophilia, that's one that they use to correct the bleeding disorder that they see in hemophilic mice, and that's kind of the basis of these therapies that are being approved for hemophilia. So that's part of the reason why we went with it, because it had been verified by our collaborators in the gene therapy group at IU. They had the design already, so then we just started to pull out different parts, like remove the polyadenylation sequence, switch up the promoters, that type of thing. And so it was more of that helped us kind of decide, okay, which parts of that genome might be more or less problematic if you get something like an incomplete genome. So you don't have all of those components. And so that's why we wanted to do it. And it was just I think it was a smart design for the study. You have, 4 groups, you want to make sure that you're not adding too many different variables. So, we're targeting these 3 pieces of the AAV genome. So it's, this group is pieces 1, 2, and 3. This group is pieces 1 and 2. This group is piece 1. It's that kind of thing. So
do you find it hard to limit the variables in this kind of study? Sorry, it just popped into my head.
No, that's a good question. Yeah, I think so. I think it's very hard because when you think of cell work, right? Cell work is very controlled, right? So you can have cells growing in a dish. You can weight the numbers. You can isolate them from different other cells, even within the same plate. You have a lot of control over those experiments. These experiments were done in mice. Can't control the mice fighting with each other. How much food do they get? The same amount of food. Especially with males. These were done in male mice. Male mice live together long enough. They hate each other. So you have to separate them out. So again, that was also a factor in this that should have put in the paper that we didn't. So I had some mice that were housed by themselves. So they gained weight faster. They were able to eat more food. So there's all these different things when you have an animal study that are very hard to control because they're living things. They're animals. So it's a similar thing if you think of like a clinical trial, right? You have all these, you have to manage those people staying on protocol. It's the same thing with the animals. You got to manage the animals getting the injections at the right time, making sure that they have the right food if you have a food restriction or a water restriction, things like that. So I definitely, in vivo experiments, it's very difficult to control all the variables.
Mm-hmm.
Emily, do you have some questions?
My questions are some similar, so you answered my question.
Oh, that's, yeah.
You were talking about
Was there?
Go ahead.
No, I was just, go ahead.
Yeah, I was wondering, was there any reason why you used the male mice specifically?
Yeah, so males are typically more at risk for hemophilia than females. So it's an X-linked disease, and males have one X chromosome. So if you have, genetically speaking, the mother of a male child could be a carrier and not know that she's a carrier, and then her child could have hemophilia. Versus if you have, a female with hemophilia, then the father would have to have it unless there's some mutation that causes it. But since it's X-linked, males was a more clinically relevant model than females.
Ready? Then Emily, any question? Do you want to add a question?
Just a question we will have. in the start. Yeah. What was the initial idea that really got this study going?
What was the? Can you repeat? Sorry.
Oh, what was the initial idea that really got
Oh, the
this study
idea.
going?
Sure. So the idea was there were some manuscripts that had been published. The main one was by, and the last author is escaping me at the moment, but I read a review because I was curious about, gene therapy and cancer. That was kind of the whole thought about what I wanted to do in this lab. And there was a manuscript where this group had reviewed hepatocellular carcinoma clinical samples, like a post hoc analysis. So these patients had passed away from liver cancer. These were biopsies of patients that had already had, disease. And what they found was that they had assessed them for presence of adeno-associated viruses, or AAVs, and they found, they detected certain serotypes of AAV serotypes. So serotypes is just a fancy name for types of AAVs. So you have AAVs 1 through 9 are your kind of biological serotypes, and then you can labs have been able to create their own basically in-house. So you have these recombinant serotypes as well. So that got me thinking about, well, what could these AAVs be really causing cancer, or is it kind of just they were there and they didn't, it was just some sort of assessment that they picked up, when they assessed the biology of these clinical samples. And then I read a few other papers about some other models where they would labs use these different recombinant AAV vectors in different mouse models of different diseases, and either long term enough in the process of the experiment, these tumors would start to rise in livers. And so, I really wanted to know, is it just the AAV biology itself? Is there something specific within within that recombinant genome that might be causing the tumors to develop? And then testing the completeness of a complete genome versus an incomplete, because there was another colleague of mine in the Shao lab who is absolutely fantastic at her research, and she was very interested in this idea of pure populations of the vectors when you make them. So that was a processing step of, we have all these components that go into making these recombinant AAVs that we can use in these studies. You don't get 100% when you mix all of the components together to get the viral genomes that you want to use for your study. So she really started to dive into, well, what if you get 50%? What if you inject your animals with X number % of complete genomes, but then there's this other percentage of genomes that don't have a transgene, or don't have a promoter, or have a mutation in the promoter? And she really was breaking down the biology behind that. And so I wanted to incorporate at a very basic level what she was doing into this manuscript, and kind of give it more of a, like we were saying, in a large, like macro level assessment of what could happen. So looking at collagen deposition, looking at immune cell populations, getting pathology reports on these mice, and kind of getting a holistic idea of kind of what was going on in these livers and spleens. And so that was kind of the idea of the manuscript from when I first joined, and kind of hopefully that was relayed in the data. But again, you always look at it and you think, man, could have done a little bit better in some of these things, and
Right.
the design, and
Absolutely. I know what you mean.
And one thing I found surprising was the incomplete vectors sometimes cause more issues than the complete functional ones. So could you explain to me why that might happen?
Yeah. So to my best knowledge, remembering. So I think with the complete genomes, what you get is this vector that is functioning as it should. So for example, if you look at, I'm gonna have a paper here. So if you look at figure 6, so figure 6 is the hemophilia B mice. So that's where, so you have two different types. You have hemophilia A and hemophilia B. Factor 9 is the clotting factor that's defective in hemophilia B. And that's why we went with the FIX vector. So if you look at panels D and E, which is the clot time. So that is so that's an experiment you can do in the lab, and you basically take you test for clotting time of platelets from these mice. So that's what we did. So if you look at the clot time of that TTR factor 9 vector, so to that second column, it's very low. That means that they're clotting, that factor 9 replenishment by the AAV vector is working. And the thing is, and it's hard to compare across experiments, but if you look at figure 3 in panels D and E, that hemophilia B factor 9 is almost exactly the same as the healthy mice. So again, what that tells me is, one, the factor 9, that vector is working, because those mice that have that hemophilia disease are clotting at a normal rate compared to the other mice. And then if you look at the other vectors, all of the vectors that don't have the factor 9 are not clotting. So they're all kind of high. So that makes sense, right? Because those mice don't have that factor. So what was nice about this experiment was that it showed that the vector was working, it was designed correctly, the mouse models were doing what they were supposed to do, and you still got an effect with the non-specific, right? So that CB human factor 9 group was still clotting low as well. So basically what I'm trying to say is that it's basically, think if you're taking a pill that's been tested and the pill has what it needs in it. Like, you have a headache, you took this pill of Tylenol and it worked because it's purely acetaminophen. Versus if you have, like the CB group or the TTR poly A where there's no factor 9, that's almost like a defective medicine, right? They're not getting any clinical benefit. So you don't know if just taking large amounts of this defective biological solution could cause things like issues with your liver, issues with your spleen. And if you overdose on the good stuff, you can still get infectious issues with that, right? So that's kind of how I interpreted it, was that the biology and the way that the vector is interacting with the cells that it wants to go into, it's functioning how it should. But when you don't have the polyadenylation sequence to help with the shuttling of the shuttling nuclear export of the RNA for translation into protein, when you don't have any transgene, so it's just a promoter that's being activated, what is that doing to those cells? What's that doing to those tissues? It could be doing anything. And that was one thing we didn't do. We didn't really fully investigate what those incomplete genomes might be doing. Are they increasing immune cell pathways? Are they, related to the collagen deposition or the difference in these cells? What are they doing? So that was kind of how I interpreted it, that you're getting this reaction because it's not the type of thing you would want to inject into your animals. And just to jump on that, so we did try and do a little bit of that. So I don't know if you all had a chance to look at the supplemental data for this. There was a supplemental package just to make it more comprehensive and annoying to read. but there's a supplemental figure 8. And if you want me to, I pulled up PubMed just in case. I can share it if you want me to. But basically what we did was, I wanted to look at macrophages, which is an immune cell that is involved in phagocytosis, responding to pathogens in your body. And so I did a staining for, let's go there, it is, for these cells in the livers and the spleens of these hemophilia B mice. And we didn't see any difference in the spleens, but we did see a difference in the livers. And that was what was kind of curious. So my assumption, or the suggestion from that, is that you are getting some sort of immune response with some of these incomplete genomes. And that fits the data with, lots of data regarding some of these AAVs, where if you overdose or something, or you have an AAV that is quite problematic to treat or problematic to deal with, it can lead to some sort of you can get an immune response, and that's when you see stuff like that in the clinic and you hear people having bad reactions to these types of therapies, it's usually some sort of immune response. But let me share what I'm talking about. Can you all see this?
Yeah.
Yes.
Great. So F480 is just the marker. It's a very general marker for macrophages. And so in livers, but in liver cells, it stains what are called Kupffer cells. And it's kind of hard to see. I think it's because of word. But you can see there's this little brown stains right here. See all the brown is here. So that's what I quantified here. And so it's this area over liver area. So you just do histology staining, stain the slides with this protein. And the idea is that if the cells express this protein, the antibody binds to the protein, you have another antibody that gives you some sort of signal that you can measure. So for this case, the secondary antibody will give you a brown color when you go through all the chemistry about how to get that activated. And so you can see, so it's control, and then it's all of the different groups. And there's a lot of brown staining when you do that chicken beta-actin only vector, which fits with the changes we saw in some of the collagen deposition, with some of the issues in the spleen. So I was not surprised, and I was happy to see that all this kind of correlated. Granted, we mentioned this in the paper, the small number of animals for this group makes it kind of there's an issue for that, but it was definitely something that was intended to be something that we followed up with. And then again, you see that vector that is complete, that has a biological effect, that corrects not corrects, but treats the hemophilia. There's not a lot of F480 positive cells in that liver. So that vector that's been designed to therapeutically help these mice with their hemophilia is not causing that infiltration or that increased staining of these immune cells. And so that was, again, a really nice to see in this paper. And again, and then you look at the spleens, there really wasn't any difference, but we did notice some positional changes. And that is something that we also mentioned in the paper. So you have kind of these regions where, again, we used F480. You have regions in the spleen where that immune cells with that marker should specifically be, and places where they shouldn't be. And then so in these dotted outlines, we kind of see, I noticed a few times that there were some animals where you had some of this kind of, it wasn't as clean and delineated as a line as in some of these. So for example, you can kind of see where you have these very large oval spots where there's no staining. And then you have all of this brown where they should be. And you kind of see some of these interesting little jutting out sections or parts where there's a little bit of infiltration. And we didn't really investigate this too much, but it was just an observation that we thought would be interesting to follow up with.
Yeah, I think so. So I'm going to pivot us just a little bit here because we were talking, before we started the recording a little bit about you making a transition from academia into a more industry position. So just for, kind of that idea of, we can move different positions and things like that, academics. Yeah. There's research involved in academics. There's also teaching involved in academics. Industry, you don't necessarily direct your own projects, but what was the initial driver for you to make your way toward an industry role from an academic role? Sure.
Several factors. One was, so I have 2 kids under 5. That was a big driving force. Just having a position or some sort of job that was and this is just from, again, my experience and how I experienced it, something to me that would be more stable. And my journey from finishing my PhD in 2017 to doing 2 postdocs and then being research faculty for about 9 months, 8 or 9 months or so, I jumped around a lot about different types of research focuses. So my PhD was in breast cancer, bone metastases, and how stress affects that process. So if you're stressed out all the time and your sympathetic nervous system is activated all the time, how does that affect your health? Again, people say, oh, stress, the phrase like stress kills or stress makes things worse. That's basically what I studied as a PhD student. And there's some truth to that. Stress, like it's that thing, stress can't really cause this, but it can make it worse. That was kind of the idea behind my dissertation work. Then I did 3 years in a multiple myeloma lab, which is a liquid tumor that, when it progresses, causes bone disease. Then I did the gene therapy group with Dr. Shao, trying to get it back to bone. And then I was in fracture healing. So I jumped around a lot because I think partially one thing is that it was really difficult for me to get my own funding. And cancer research has just been, it's incredibly competitive to get that money. There's a lot of money, and that's kind of the funny part. There's a ton of money for cancer research, as there should be, but it was very hard to get it for me. So I loved a lot of aspects of the research. I loved teaching. I have a certificate in college teaching from my time as a PhD student at Vanderbilt. That was one of the most rewarding, funnest things that I did to date for my career, my time as a scientist. So I was able to kind of do the teaching, do the research, but it was really hard for me to kind of see where I was going. And it just always felt like the goalposts were moving, right? So it's like, if you do these things, then this is what you'll get. And then it was, just kidding, we're going to have to do these 3 other things so that you can get to this spot.
Yep.
So at some point, for me, it was, what do I like to do? I had a daughter and I had another one on the way. I just needed a change, and I needed a change. And so I looked at jobs at different companies. I looked at different positions, and I got this position that I have now, and it came at the right time, and I've really enjoyed it. But it was not an easy transition, because I really did feel like I wanted to be a professor running a lab, teaching students, doing this type of stuff all the time. Helping students kind of figure out what what science projects do they want to look at, what interests them in a lab. But it just wasn't, it was basically not getting the grant, not getting that 6-7 figure grant to fund, a program that we could build a lab on. And
I got to say, just from this conversation, I think you really have a talent of explaining what you're trying to get out really well. No, I mean, sometimes, that's one of the criticisms I think we get as scientists, researchers, is we're not able to express those complex ideas into a simple way. And I think you are doing an absolute fantastic job, at least here, doing that. I've been just blown away, because some of this stuff, it's deeper into the genetic side of things than I look at as more of a phylogeneticist person. And you're helping me reconstruct how I go over it, and you're absolutely, I think, gifted at explaining this stuff.
Well, thank you. I appreciate that a lot, because it's definitely something that I noticed as well, that you have this. I kind of was thinking about this. I had a friend of mine in grad school who hated the show Big Bang Theory because what he thought was, he's like, they basically say all scientists are these types of people that, like you said, can't communicate, or awkward. Mm-hmm. And it's not that there's not some truth to it. I think everybody's awkward. Everybody has some tick or whatever that makes them weird. But yeah, I think that's one thing, if I could, relay something to students, is that figure out ways to improve your communication, written, verbal, all that stuff, because it helps you and become more comfortable with presenting, with speaking, try and just put yourself out there to find ways to talk about stuff. I think for me, journal clubs, so when you read these, like you are doing, reading these papers, talking about it, that was one of the best things that helped me get to that point where it was easy to talk about things. And then it's another way to learn. You learn about all these different fields that aren't, my area of focus. And that's also been helpful in the transition out of the academia to industry, because with industry, with academia, you focus in on one thing, right? Like this is you want to study this, so you have to become an expert in this. When I transitioned out, my expertise in cancer biology was useful, but it wasn't what got me hired, if that makes sense, right? I didn't need to show that I'm an expert or I have a 14 years of experience in cancer research. That wasn't what got me into the job I have now. So.
And what has been the most rewarding aspect regarding the scientific aspect of it all?
Of my time, you mean my time when I was in gene therapy, or just generally?
Just generally.
I think what I love about it is I love just learning about things. I knew what gene therapy was, but I had zero experience. I had never done molecular cloning before. This is something that, you all as undergrads, if you go to some lab and say, okay, let's do some molecular cloning, find these enzymes of those guys. I had never done that. And you have all these students that, were much younger than me that have cloned vectors and made genes. And I had never had that experience. And it was humbling, but also it was good because it was challenging, and it was something that was, that I saw as, if I really wanted to stay in research and academics, I needed to understand it. I needed to know how to do it before I get to the point where I'm delegating anything, right? Because when you get to run your own lab, it's, the first couple of years you're doing everything, right? You're managing it, you're running experiments, you're talking to your students, but then there's a time, and I saw this with my mentor when I was getting my PhD, is that he had this big transition away from being at the bench, being involved in the experiments, because he had administrative work. He was running, our bone center at the time, and our lab was very large. So he didn't have the time to sit with me and say, okay, we're going to run this animal study. When are we sacrificing your animals? I can be there at this time to do it. It was very hands off. So I wanted to learn as much as I could. And then the other thing that was fun about, at least in this lab, this is where I did the gene therapy, is that I was one of 3 people that were hired at the same time. We were all postdocs, and we all had very different expertises. I was the animal biologist. We had someone, she was a chemist. And then this other researcher, he was very good at molecular biology and experimental design. And so all of us coming in this lab together, where it was Wei Dong, who is still to this day one of the best bosses I've ever had, and his research scientist in the lab, we were so efficient. We balanced each other out. We helped each other. I was able to provide this detail while they were able to provide this aspect that I didn't know about, and we learned from each other and we helped each other.
The true idea of collaboration.
Exactly. And I've been plugging this book for the last month. I read this book called Surrounded by Idiots. And it's a great title. The title grabbed me and I read it. And it's basically about how you communicate and how different people communicate and how you're perceived and your actions. It's a behavioral book. It was really interesting. And the message behind that book was, so I'm a yellow, there's 4 colors, right? And I'm this yellow, and you don't want to be in a group of all yellows because you won't get anything done. You don't want to be in a group of all blues or all greens because you won't get anything done. The teams that get things done are where you have mixtures of all the different types of people. And that was what Wei Dong's lab was. We were all different. We were all brought our own strengths. And it was, like I said, it was the most productive, paper-wise, and I learned the most in that 3 years I was in his lab. And I think that was what drives me, is this idea of learning and just understanding and curiosity. That's just what it was. And I'm still doing that now, even in my new position. Learning a whole new set of things behind science and how it translates from what is done in labs and in animals into humans. And so it's been it's a great,
awesome.
You got to be curious.
Yeah.
You got to be curious, and have a reason to want to put the effort in and do the work.
Absolutely. Emily, do you have any thoughts on this stuff?
No.
Nothing?
Well, I have a question.
Oh, good.
Sure.
It's more personal. We have motivation for choosing this career. I want to know what is or who is your motivation to choose this career?
It's a great question. I don't know. I think it's just that. I think it's cliche, but it really is just my family. It's just having something that's stable. I think I've realized that one of my fears is being unemployed, is not having a job, not having a steady income, not being able to just have that. All right, I have, sometimes it's, 9 to 5 is kind of, it's an older thing. It's not, the 9 to 5 doesn't exist 5 days a week. I have something that is stable. But I was always one of these people, and I don't know if people have done this, you all are like this, but I didn't really ever have a scientific person that I aspired to be. I had people that I was in awe of, that I really enjoyed the research. There's a guy, if you're ever bored, a guy named Ralph Adams, who was at Max Planck University out in Germany. His research is, if you ever get a chance, read his papers. He got really beautiful pictures. His pictures are prettier than mine from this paper. But I just was never one of those people that, had scientists that I aspired to be. I really just wanted to be the best at what I could be. And then when my wife and I got married, and we met in graduate school, so she has a PhD as well, and she was in research. And then we had our 2 kids. It just became about, I can still make an impact having a job that's stable. I don't need to make an impact as a professor. I can still do things like this. I can still talk to students and teach in different capacities in my new position. And that can be enough for me at this moment.
Very cool. That actually transitions us really well into our wrap-up questions that we ask everybody on the podcast. We have just 3 questions we kind of ask everybody, and I think that transitions really nicely. One of you two want to start us off there on the wrap-ups?
Yeah, I'll start it off. What's one piece of advice you'd give your undergrad self?
Oh my gosh. Yeah.
That you'd listen to, maybe.
Yeah. Yeah, right. Don't be intimidated by the fact that you're surrounded by smart people. I came in, I was a top 10 in my high school class. I did honors classes, I did all that stuff. And then I got to University of Wisconsin, and everybody was that. And I got intimidated. I think the first year I wasn't intimidated by it, because it was just, you're figuring it out. But by sophomore year, I was very intimidated. Started to question whether I should be a molecular biology major. Should I continue on in this path, or should I do something different? And I think I would tell myself that you don't need to be intimidated by everybody. You got in for a reason. You just have to put in the work and figure it out.
Absolutely. I have experiences from my undergrad where I feel like, hey, even though I think these guys are smarter than me, I'm in the room too.
Bingo. Yep.
What's something more people should understand about scientists' or researchers' process?
You don't have to go to medical school if you're a biology major. That is kind of one of the things that looking back, just irks me so much. And it's not because it's about getting the MD or DO or that profession. It's the fact that if you're smart, if you're in honors classes and you're a science major, the automatic assumption is that you're going to go be a physician versus a researcher versus a writer versus a veterinarian versus there's so many facets of society that would benefit from people who understand science, scientific method, critical thinking. I wish it's undergrad institutions, right? That they would expand out beyond just saying, okay, you're a biology major. you're going to be pre-med, that having that question, I think you just that jump has to stop. Has to be an option, but you can't exclude all these other things that you can do with a science degree.
I think I've talked about this before with some students in a sense of, the career options that I think people who get into biology think exists are only those medical options or what they think are lesser paying but really fun, park ranger positions, and those are the only two jobs they really see, and then of course they consider a professor in there too, but those are the only career paths that they see until you start telling them things like, every chapter we're covering in here are 3 different career paths. In this chapter we're reading in this very specific genetics textbook, every chapter has 3 career paths that you could go down.
Yeah. It's either having professors like you that know that and are taking the time to do that, or having that system set up where you have those career resources where that is something that is discussed. I remember we use the term loosely, a advisor at Wisconsin. I met with him my junior year, started junior year, because I'd had my sophomore year was, relatively speaking, terrible. I had one bad semester, and I was, in that funk, right?
Right, I've been there.
He said, he's like, okay, your grades got to get better. And I'm like, well, of course. I mean, what else are you going to tell me? I remember him telling me that.
Yeah, that
and I realized,
yeah,
and I remember her just thinking, he didn't look at anything that I had. He looked at something 5 minutes before I got there, and I need to figure, again, going back to that idea, I need to figure it out on my own. I need to talk to some of my biology professors maybe, but I have to figure this out on my own. And luckily I did, finished with a 3.5, which is not terrible, but it was not a 3.5 going into junior year. So it was
honestly, it almost sounds like we had similar undergraduate journeys to some degree.
But it wasn't just me.
No, I mean, we both finished in Wisconsin too, which is fun.
Nice.
But we got one more question for you.
Sir.
So,
if you weren't doing this, what would you be doing?
Oh my gosh, that's a great question. I had a weird love of geography when I was a kid. And I still say one of the best classes I ever took at my undergrad institution was a European geography class. And I remember thinking the professor was awesome because his job, basically, he traveled all around Europe, documented it, and then wrote a textbook. So I would do something like that where I could just be this. I think that type of history of, like how the books like how Guns, Germs, and Steel, or how the states got their shapes, or some of these other types of books where you just talk about how land and countries and things have formed and the history behind that. I think something like that would be super fascinating to be, knee deep in and kind of explore on that. But I would probably do that and maybe be a college professor in that realm versus science. But I would say those are two really good books. Highly recommend reading those as well.
All right. Well, thank you so much for taking the time, Dr. Mulcrone, and thanks to our guest hosts, Emily and Alfie, for taking the time to put together the questions, find the paper, and then the sit and wait process of getting everything together, because I'm sure that gets everybody going a little bit. But thank you so much,
sir. Thank you.
Of course, I appreciate it. Thanks for asking, and you two did a great job, and I'm glad that I could clarify some things on the paper. And if you have any follow-up questions, my Gmail account, I'm happy to follow up with any questions that didn't get asked or that come up later on. And I was happy to be asked and to be here.
Yes, sir. Thank you.
Yep. And this has been STEMpaths. I've been your main host, Dr. Thornton Larson. Thank you, and we'll see you guys next time.